Okay, so we are going to go over some notes that we took today on a new type of cell division called meiosis. I'm going to go ahead and share my screen here. In 1 second. There. Oh, okay. So you have a hard copy of these notes currently in Google Classroom, and so I figured I'd give us a little bit of a walkthrough. So we obviously filled these out together. You got these all filled out now for yourself, so you can either print them out or get a hard copy when you return, and just reference these notes as needed while you're gone. So up until now, we've talked about only one type of cell division called mitosis. And mitosis is a type of cell division that's used to create all of your body cells except for one, your sex cells, or what we call our gametes. The goal of mitosis is to essentially make a clone, a copy of the original cell. And we remember from our test on mitosis, you have one cell that splits in half, divides, and becomes two new cells. But those two new cells are the same as the original cell. Meiosis, on the other hand, has a different goal. So the goal of meiosis is to make your gamete, your sex cells, and our goal is to create cells that have half the amount of genetic material needed in order to create a person. As we know, humans have 46 chromosomes. That's where our genes are located, and they're found in the cell's nucleus. And so after meiosis, if you're female and you're producing eggs, your egg cells will have 23 chromosomes, and if you're male, your sperm cells will have 23 chromosomes. So that during sexual reproduction, sperm and egg unite, we get the 46 chromosomes that make a human being. So after meiosis 2, we'll get an idea as to why each organism inherits a single copy of every gene from both parents. So why you receive half of your genetic information from your mom, half from your dad. And well, each of our gametes or the sex cells have one complete set of genes. And we'll also see too why all of us are different from each other. Why if you have siblings, you're different from your siblings, even though you have the same parents. So there's a couple vocab terms just need to understand first before we start the notes. So we know what a chromosome is, but we have a new description though for the chromosomes involved in meiosis, and they're what are called homologous chromosomes. So these are chromosomes that are two sets, one that you receive from your father and one that you receive from your mother. So they contain the same DNA sequences of the similar genetic information, but half of it's from your mom, half of it's from your dad. We also have the term diploid, which means double. So a cell that contains both sets of chromosomes. So for humans, diploid cells are 46. And then haploid, which means half. So meiosis is going to make haploid cells, so cells that have half the number of chromosomes as they would diploid. So for people, it's 23. And that N there is there to tell you the number of chromosomes in a gamete. So if it's 2N, then that means it's diploid. So double what it would be for mitosis. If it's haploid or N, then it has half the amount. And so that's a indicator that it's a gamete or a sex cell. So in meiosis, we go from diploid cells, so cells that have both sets, to haploid cells, cells that have half a set. Now, the steps of meiosis. So we have more steps than we do with mitosis, but the nice thing is now that we have an understanding of mitosis, we know the order of these steps, and we have a general idea of what's happening at each step. So meiosis has 2 rounds of division. And so since there's 2 rounds of division, we indicate those steps by round 1 and round 2. So meiosis 1 has an interphase, prophase, metaphase, anaphase, and then we just kind of group together just so things aren't so long, telophase and cytokinesis, so the splitting of the cytoplasm. And after 1 round of division, we go into a 2nd round of division, and again, the steps look fairly similar to mitosis. Now, they're not exactly the same, so we're going to talk about those differences. That's why we get different types of cells than we do in mitosis, but how they look, though, are very similar. So we're going to start off with the first round of division, meiosis 1. So just like in mitosis, during interphase, before we divide, each chromosome is going to be replicated or copied to make that X shape, just like in mitosis. Now, during prophase, the organelles for cell division are going to be made, and they're going to go into their proper place, but we're going to have something different happen than what happens in mitosis. So here we have a cell that has 1, 2, 3, 4 chromosomes. This is the diploid number for the cell. So during prophase, the chromosomes are going to pair with their homologous chromosomes. So the one chromosome, let's say this is on this chromosome here, we contain information for hair color and eye color. So the genetic information for hair and eye color that we got from mom and the one from dad, they're going to pair up with each other because they contain the same set of genetic material. And so the pairing of homologous chromosomes, they form what's called a tetrad. So that's just a group of 4 chromatids or 2 chromosomes that make up pairs with each other. Now, when they pair up, something very important happens, and that's what causes changes in genetic information for everyone during sexual reproduction. That's why if you have siblings, you look different than your siblings, and this is what's called crossing over. So as these homologous chromosomes pair up, they're going to rub against each other. They're going to be very close to each other while they're in a pair, and as they pair up, they're going to exchange portions of their chromatids in a process that we call crossing over. Now, it's the same sequences of DNA, so nothing bad, nothing detrimental happens, but here's where we do a swap then of genetic material. So let's say on this chromosome, we have the genetic code DNA sequences for brown hair and brown eyes. Say we got that from dad. On this one, we have blonde hair and blue eyes. We got that from mom. So they're going to buddy up, cross over, exchange portions of their chromatids, portions of their genetic information. And so now this chromosome that originally had sequences to code for brown hair and brown eyes now might have brown hair and blue eyes. And this one that originally was blonde hair, blue eyes, might now be blonde hair and brown eyes. So it's a little more complicated than that when it comes to eye color and hair color, but just to give you a kind of a gist of what's going on. So we go into metaphase. So remember, metaphase, middle, the chromosomes are going to line up in the middle of a cell. So another thing that's different in meiosis 1 is how they line up during metaphase during the first round of cell division. So in mitosis, they line up one on top of each other, so 1, 2, 3, 4, during metaphase. In this case, the chromosomes are going to line up with their tetrad pairs. So instead of being one on top of each other so that each side of the cell would get 4 chromosomes, they line up with their tetrad pair. So then during anaphase, the spindle fibers pull apart and the chromosomes move to opposite ends of the cell. And because they lined up as tetrads instead of one on top of each other, once we get to telophase, where the chromosomes each go to their own cell and nuclear envelopes start to form around them, we end up with 2 cells that are now haploid. So they have half the number of chromosomes that the original cell started with. So in our first picture in prophase, our single cell had 1, 2, 3, 4 chromosomes. Our 2 new cells now have only 1, 2, and 1, 2. So now we have haploid cells. So now we're going to go through another round of division, and here it's going to look just like mitosis. So we go right into prophase. There is no interphase at this point, so there's not going to be any replication of DNA. We're just going to divide again. So during prophase, the chromosomes head to the center of the cell. The spindle fibers form again. They go to their correct spot, and the chromosomes are now going to line up one on top of each other right in the middle of the cell, just like before, once we get to metaphase. And then at metaphase, they're all lined up. After metaphase, we got anaphase, so the 2 chromosomes separate from each other. And then we end up with 4 cells, all with a haploid set of chromosomes. So each one of these has 1, 2, 1, 2, 1, 2, 1, 2. So these last couple slides here on the back are just a quick side-by-side comparison of the 2, as well as what each round of meiosis looks like. Just on a single slide, so it might be a little bit easier to look at than going all the way through. But again, our final goal here with meiosis is to complete daughter cells. And in this case, with meiosis, we end up with 4 daughter cells instead of 2 at the end that are not genetically identical and that are haploid, so have half the number of chromosomes. Whereas with our goal of mitosis is to make 2 cells that are genetically identical to the parent or to the original cell. So it's a quick walkthrough of the notes. Attached to this assignment, the makeup assignment, is a practice that we did in class. I also have the key attached so that you can check your understanding. But if you still have any questions after watching this video, looking through the notes, reading through your book, etc., feel free to just send me an email, just so you don't fall behind here. But hope you're all doing well, and I'll see you all soon.